Historically, early zoologists assumed birds possessed a primitive, unrefined sense of taste and smell, or lacked one entirely. That assumption is incorrect. Avian chemical sensation actually relies on two distinct parallel physical pathways, chemesthesis and olfaction. Chemesthesis is the specific detection of irritation and pain. Olfaction is the detection of true odor. In practice, a strong external chemical stimulus often triggers both systems simultaneously. Avian sensory architecture represents a strictly divergent evolutionary path, engineered for specific ecological pressures. To understand how this works, we have to trace an environmental chemical signal from the exact moment it enters the bird's airspace, all the way down into the genetic code that dictates the animal's reaction. Reverse engineering this organic sensor suite reveals how microscopic protein mutations dictate the survival and foraging behavior of an entire vertebrate class. Chemesthesia acts as a subcategory of nociception, the physiological perception of pain. When triggered by a chemical, it initiates a reflexive withdrawal sequence that does not require the bird to learn avoidance over time. The macroscopic hardware driving this pathway is the trigeminal nerve, or cranial nerve 6. Its ganglion splits into the ophthalmic, maxillary, and mandibular branches, innervating the beak, eye, and nasal floor. The transmission lines relaying these signals are unmyelinated C-type polymodal nociceptors. These afferents carry impulses at conduction velocities of 0.3 to 1 meter per second. This schematic details the peripheral terminal's transduction cascade. Sensitizers like histamine bind to receptors, initiating an intracellular cascade to TRVP1 and TRPA1 phosphorylation sites. The signal then travels via anterograde transport to the cell body in the dorsal root ganglion, altering the neuron state. Here we see a stark physiological divergence. When mammalian sensory afferents are exposed to irritants like capsaicin, they release a neuropeptide called substance P to trigger pain. Avian afferents do not release substance P under the exact same exposure. The lack of substance P release proves the underlying neural hardware is highly conserved across taxa. The chemical triggers that successfully activate it, however, are radically different. The actual mechanical triggers for the pain response are transient receptor potential, or TRP channels. These are non-selective cation sensors embedded directly in the cell membrane. This diagram displays structural models of TRP channels. In mammals, the TRPV1 channel on the left is highly sensitive to capsaicin, the active heat component in chili peppers. Birds possess their own version of TRPV1, sharing a 68% genetic identity with the rat equivalent. Yet the avian version is functionally insensitive to capsaicin. A precise molecular mutation, substituting glutamic acid for alanine at position 578, prevents capsaicin from locking into the binding site. Instead, the avian system relies on TRPA1, identifiable by its unique 17 anchorin repeats extending into the intracellular space. Avian TRPA1 is highly reactive to specific compounds that mammals largely ignore, such as methylanthranilate, which is found in corn grapes, and pulegone. When one of these target molecules successfully binds, the channel pore opens. This action allows a rapid influx of intracellular calcium that fires the nerve. This single amino acid substitution dictates which chemicals an entire vertebrate class will perceive as painful stimuli. This drastic behavioral divergence between mammals and birds is governed by strict molecular geometry. This graph compares the food intake of rats versus starlings. When exposed to capsaicin versus veritrolamine, the tolerance levels perfectly invert. To calculate the exact chemical rules of avian repellency, scientists use quantitative structure-activity relationships, or QSAR.
This plot illustrates the relative reduction of fluid intake, isolating highly aversive compounds like acetophenones and anthranolates on the far left. For an exogenous chemical to trigger the avian pain response, it must meet three specific physical prerequisites: an aromatic heterocyclic core, a high degree of basicity, and high electronegativity. High lipophilicity is also mandatory. The compound must be able to efficiently dissolve into and penetrate the lipid bilayer of the cell membrane to physically reach the receptor pocket. The electrostatic forces are so precise that 94% of the variance in a bird's aversion response is dictated by parameters like polarizability. Polarizability measures the relative susceptibility of the molecule's electron cloud to be distorted by an external electric field. This avoidance response operates on the physics of molecular shape and electron cloud distortion, rather than a subjective experience of flavor. The chemostatic pain response only covers half of the semsome. The parallel route is true olfaction. Air currents move over the invaginated nasal conchae, directing odorants straight down to the olfactory epithelium cilia at the caudal most chamber. The initial transduction cascade begins when these odorants bind to 7 transmembrane G-protein-coupled receptors, altering their conformation. This conformational change causes a rapid chemical chain reaction. Adenylyl cyclase 3 activates, forming cyclic AMP, which ultimately generates a localized depolarization of the ciliary membrane. These cross-sections compare rat and pigeon olfactory bulbs. While avian layers appear less distinct, fundamental cytoarchitecture is conserved. Axons expressing the same receptor type converge into spherical glomeruli, which pass signals to the mitral cell layer. The mitral cells project this process data directly to the piriform cortex, allowing the bird to achieve higher-level cognitive integration of the odor. The genomic capacity supporting this network dismantles the myth of the microsmatic bird. Birds possess up to 11 distinct olfactory receptor gene families, with specific lineages showing a massive expansion of the OR14 family. This rapid genetic expansion provides birds with a highly complex, airborne chemical detection network tailored precisely to their ecological niches. When we connect the internal molecular data back to the macroanatomy, we see exactly how hardware limits dictate behavioral capability. These 3D models of diverse avian brains highlight the extreme physical variation in the size and shape of the olfactory bulbs. This table summarizes the mean ratios of olfactory bulb diameter to cerebral hemisphere diameter. Species with larger ratios, like the massive 29.1 seen in seabirds, possess low chemical detection thresholds that rival macrosmatic mammals. In the real world, a high-concentration volatile stimulus rarely triggers just one pathway. Through retronasal stimulation, it activates both the olfactory nerve and the trigeminal nociceptor simultaneously. The concept of flavor is simply the ultimate cognitive integration of these two parallel pathways. TRPA1 channel firing and mitral cell firing travel upward, fusing together in the higher brain at the exact same time. We observe the ecological consequence of this integration in the wild. European starlings utilize specific aromatic plant volatiles to fumigate their nests against parasites, a complex behavior strictly mediated by olfactory discrimination. We also see the evolutionary consequence. Pepper plants specifically evolved capsaicin to exploit mammalian TRP channels for defense, while completely bypassing avian TRP channels to secure a reliable, pain-free seed dispersal network. The avian sensome persists as a parallel sensory universe, governed by a molecular logic distinct from our own.